Hello everybody, I'm Alex and welcome to the second in the Triverity Laboratories webinar series. Today's subject is analytical development and we will provide an overview of what underpins product quality from early development to commercialization. Please submit questions at any time using the chat panel or the Q&A panel, and these will be addressed during the Q&A session at the end. All participants will be muted during the presentation. This session will be recorded and we will share it with you afterwards. If you experience any audio or connection issues, please refresh your browser or rejoin using the webinar link. We appreciate you taking the time to participate and hope you find the session informative and useful. With that, I will hand you over to Wendi.
Thanks, Alex. Hello, everybody. Thanks again for joining us today to talk about analytical development. It is a subject that's near and dear to my heart. I've been doing analytical development of pharmaceutical products for over 20 years. This is, as Alex said, the second in our series. The first one was specifications for pharmaceutical products. Analytical development is going to, this presentation is going to be kind of an overarching view of analytical development. There's going to be a shockingly few slides on method development and validation, and we'll get into those in other webinars. So an overview of what we're going to talk about today, we're kind of breaking analytical development up into initial assessment, method development, characterization, method validation, late stage development, and then the sections of the CMC that are applicable to analytical development, and then wrapping up with life cycle management. There's a few applicable guidances. I didn't put the CFR references, 21 CFR Part 211, on here because there's nothing that really speaks to analytical development just as a whole for product release and stability and all of those kinds of things, characterization of impurities and contaminants. So I left that off this time, but there's a number of USP chapters and ICH guidances. So USP 1225 for validation of compendial procedures, 1226 for verification, 1224 for transfer, and then a whole bunch of chapters for the specific contaminants and whatnot. Then there's also the ICH chapters, the big one, ICH Q2A, which is analytical validation, as well as Q3A and 3B for impurities, and that's actually going to be our next webinar going over organic impurities in pharmaceutical products. And there's a new draft chapter for extractable leachables, which is Q3E. We're going to talk about that a little bit. And then the pharmaceutical product life cycle, Q12 and Q14 for analytical procedure development. So analytical development is a holistic approach to product characterization. It's specification setting and methodology. It brings together product knowledge, R&D data to design robust methods and specifications. But really analytical development goes beyond just the specifications. It's all the work that goes into making sure that you understand your product and that you are ensuring that the product that goes to market is consistent and safe for use. So again, analytical development is broken out into these stages. The method development, we do the initial assessment, and we're going to walk through all the stages of this. Initial assessment really is a lot of just data gathering. Method development happens once you have a rough formulation or you have an API that you're working on. But product development really happens under the method development and the characterization. Once you get to method validation, your formulation should be set, and then late stage development, also your formulation should be set. So initial assessment. Again, data gathering in the initial assessment. I would always create a Word document and put all of the information that I had. What is the API? What are the strengths that we're looking at? Impurities, any USP information, all of that, kind of gather it all together. We're going to walk through each one of these separately here. So the first is sourcing of the API. A lot of times this has happened in procurement or supply chain. They're figuring out who manufactures the API, and then but the technical review needs to be done by somebody that has knowledge of the requirements of an API. So typically when you're sourcing an API, you get the tech package, which is the open part of the DMF. And it is the non-proprietary sections of a DMF that the manufacturer, the API, is willing to share. So with that, it has things like a description of the API, the methods for maybe assaying impurity, maybe a residual solvent method in there. Talks about things like particle size or polymorphism. There's batch history. You want to look at that and see if the batches are consistent. How many have they made? How far apart were they? Looking at impurities in there. How much information are they providing on impurities? Like what are the process impurities? What are the degradants? And then there's specifications. If it is a compendial API, you want to look at that specification against any compendia that exists. And then look at their stability data. How many lots do they have on stability? What are the trends in there? Do you see any degradation or any red flags in that stability data? And also then the supplier history. Have you worked with that supplier? Are they reputable? Do they have a good history with you? And then finally, the GMP status. Has that DMF been filed with the FDA? If it has been filed with the FDA, has it been reviewed by the FDA yet? If that DMF has been reviewed by the FDA, then it gives you kind of a leg up because they probably have already gotten a number of deficiencies and have addressed those. So it kind of gives you some assurance that when your application gets submitted, then it's not going to, there probably won't have that many deficiencies at that point. Most things have been addressed. The next is excipient sourcing. And with excipient sourcing, again, that happens a lot in procurement. It happens probably in formulation development, but analytical development wants to get involved. You want to identify the critical excipients, take proper care in sourcing those, assess the potential suppliers, review the excipient information package, which is similar to, it's much smaller, but it's kind of like a DMS. So it's general information. It'll tell you the country of origin of the material, the GMP status, any regulatory information like BSE, TSE statements, or elemental impurity and residual solvent statements, and then also provide allergen or GMO or halal and kosher statements. Also can provide like nutritional and organic certification, but if it's critical that you're able to get at a minimum the BSE, TSE, the elemental impurity, and the residual solvent statements. And then you want to evaluate those C of A's or the specifications against if there's a compendia, hopefully there is, and make sure that they are meeting the compendial requirements. There's also some excipients that have different grades. So there'll be a pharmaceutical grade, which is like USP, or there's some that also are could be cosmetic and food grade as well. And you want to try to stay away from those and get the pharmaceutical grade if it's available. Impurity assessment, again, we're going to do the next webinar on this. So this is a shockingly small discussion on assessing impurities, but you want to look at the drug substance impurities, look at the DMF. What do they say is are there the starting material, the intermediates, what are the potential of process impurities, and then also looking at any degradants that they've identified already. Then look at the USP or EP for if there's a monograph, what are the impurities that they have in there? Do they correspond to the impurities that are in the DMF? And then also looking at literature. So doing a literature search to see, are there methods out there? Is there any data on the degradation profile of this API? Elucidation of structure is something that I think sometimes happens at the end, but maybe should happen at the beginning. So in the application for the drug product, you need to provide information on the elucidation of structure of the API. So you need to give scientific evidence that supports the molecular structure and formula, as well as polymorphism and stereochemistry. A lot of this information is provided in a DMF, but you probably want to confirm at least some of it, and if there are gaps, generate that data. Sometimes if you wait too long, it takes a while. It can take a while to get some of this data. And then 2, if there's any issues with it, it could delay your filing if you're not getting it early enough. And then you want to check the compendial status of your API and the drug product. So look and see if there's a monograph available, whether that's in the USP or the EP. If there's no USP monograph, then FDA often looks to the EP. So the drug substance, you want to look for drug substance, drug product, and then excipient monographs. You want to look at the ICH and the USP test requirements for your dosage form. And then if there is a monograph, you want to look at the applicability of the tests that are being provided. A lot of times the method for impurities isn't going to be specific for your impurities. So the USP monograph was built usually on 1 or 2 different API sources. And if there's a different synthetic pathway or there's other impurities that have shown up, then they might not be included in the monograph, which isn't a big deal if the method is still specific to those new impurities. If the method isn't specific to those new impurities, the method really isn't appropriate. You also want to check the sensitivity of the method. We've seen a lot of USP and EP methods not have the sensitivity to get an LOQ that meets ICH requirements. And then just a quick conversation on quality by design. So quality by design is a systemic approach to development, and it starts with the quality target product profile, the QTTP, which is the dosage form, the strength, and if there's any release profile that you need to have. From the QTTP, then you get the critical quality attributes, and those are the tests and the criteria that the product has to meet to be able to meet the quality target product profile that you put together. Following on that then is the analytical target profile. And the ATP is the requirements for the method to be able to test for the critical quality attributes. So things like the range of the method, what impurities do you need to have, what sensitivity do you need to have so that you can see low-level impurities. Now we're going to talk a little bit about method development. So your analytical method strategy, your method needs to be appropriate for the specification, and that's really where your ATP comes in. It needs to be phase appropriate, whether the method needs to be qualified or validated, and then sufficiently developed early. It's really important to have your method, it's kind of like the chicken before the egg sometimes, the cart before the horse. So you want to develop the method early enough so that you have data, the data that you generated early in development still is applicable and can be used later in development. As an example, if you use one lot of a raw material that you find out later was really critical in early development, but your methods weren't good enough to really rely on the data that you generated there, you've kind of lost out on an opportunity to create a lot more robust of a data package. So doing some work early to understand, and that's where the data gathering comes in, doing the work to get a decent method started so that that data can be followed through all the way to the end of development. The analytical target product profile. The goal of the ATP is to attain an analytical procedure that's fit for the intended purpose, to measure an attribute, which is to measure an attribute or attributes of the material with the needed specificity, accuracy, and precision over the reported range. So again, the QTTP feeds the CQA, which feeds the ATP. And really the ATP really forms the basis of the analytical procedure. So a lot of this, that's kind of why I had that in the initial assessment. Analytical development should be involved in determining the QTTP and determining the CQAs. And then before a lot of method development happens, then they're figuring out what the analytical target product profile is, or target profile is. That's something that could change as you learn more about the product. certainly as you identify new degradants, or you have excipient changes, things like that, or you realize that there is a component to your product, maybe like viscosity, that is critical to drug release, and therefore it's something that needs to be measured a little bit closer. Method development, and this is focusing on like a assay or impurities method. So you want to start with, what are the method requirements? What are the analytes I need to measure? What are the ranges I need to measure it in? How sensitive does it need to be? You want to find your starting point. The technology is the methodology, or the instrumentation you're going to use. So HPLC, GC, maybe it's a dissolution method that you're working with or developing. You want to come up with your basic instrument parameters. Those can come from previous experience. They can come from literature. They can come from a DMF or a USP monograph. So you want to try to find a starting point for your instrument parameters. Sometimes there's just nothing exists. And we start with 90, 10 organic, or aqueous to organic to 1090, and just run it and see what happens. My favorite part of method development is the first injection on the instrument for a new API, just to see, is it going to lute where I think it's going to lute? What is it going to look like? I get pretty excited about that. Then the next is the initial sample preparation. You want to start with method development with as kind of comprehensive extraction as you can. So you don't want to do any SPE, or you really just want to kind of dilute and shoot as best you can. It really depends on your sample, but you don't want to exclude anything from the start. Keeping in mind that you also don't want to gum up your instrument. So it's kind of a give or take there. Once you have a starting point and you're doing your first injections, then you want to start assessing your matrix interference or any matrix effects that you might have. So looking at a vehicle of or running the excipient separately. You want to assess the sensitivity and the LOQ. What can you achieve? And sometimes once you start looking at the LOQ, then you have to adjust your sample preparation, maybe increase the sample preparation to be able to get your LOQ. And then looking at precision, I typically do an N of 3 of the drug product and the API, just to see, can I get consistent results with my sample preparation? We don't typically see a lot of variability in the instrumentation. If you've got things developed well enough and your instrument is running well, typically precision issues come from sample preparation. And then looking at spike sample accuracy, you always have to take spike samples with a grain of salt. The API isn't incorporated into the matrix like it would in the product, so it's a lot easier to remove. If you're lucky, like I was, you'd have a formulation group that can make you samples that are formulations that are at different concentrations. So we would get formulations at the 100%, the nominal value, and then 50% and 150% to evaluate the accuracy across the range of the method. And then once you kind of have your method developed, relatively developed, then you want to look at system suitability. What is your system precision, and how do you want to set that? What criteria do you want to have? The asymmetry, tailing factor. You want to look at plates and sensitivity, which is like the LOQ, the signal to noise of the LOQ, if you can consistently get 10. If your LOQ standard that you're running in your system suitability is at 10 or 11, you probably want to look at a method change to increase that a little bit, so you're not right at the limit there. And then also looking at standard accuracy. We typically do an A-B standard. The A standard is what we calculate against. The B standard is our confirming standard, to make sure that the A standard was prepared correctly. So you want to go through and set your system suitability, kind of your plan for system suitability, but then also the criteria that's going to go into that. Then there's a couple of other studies. One is robustness, the other is force degradation, that we do as a part of method development. Robustness can be done as a method development tool. You can use design of experiments. So it really is just taking the methodology and making small changes to it. So changes in things like injection volume or gradient time, mobile phase concentration, maybe the pH of the mobile phase, things like that, to see how those small changes affect your results. Typically looking at system suitability, specificity, those kind of things. So you want to do, you can do this in development to help you narrow down your chromatography, but then it's something once you have it set that you want to do to show that you're not on the knife's edge with anything, like the signal to noise of the LOQ. If you're changing the injection volume by 10%, are you going to be failing your LOQ? Those kind of things. And good robustness studies, they're called out in ICHQ 14 as part of the analytical procedure development, and it can be used to help you reduce the burden of changes post-approval. So change something that is a CBE 30 maybe to a CBE 0. So that's something to keep in mind, that the more work that you're doing in development, the better the data package you can provide, maybe the less burden that you have later. And we've all lived with the post-approval method issues. Like, we had we've had ones where, all of a sudden we have a new impurity that shows up. One was a UV protectant coming through the label, and all of a sudden we've got this massive peak because we didn't have that label on stability. So as soon as we made the first product, we saw that. So there's always some method changes that have to happen post-approval, and the more work you can do during development and during validation, the less that burden is going to be. And then also a good robustness prevents failures during method transfers. If you can look at different instruments, multiple columns, age of column, all of those kinds of things really help make sure that you're not going to have problems down the road. We had one situation where we developed a method on a column, and it had been used for a product with a surfactant in it. We did the API development on that column. It's the same API that was in the product with surfactant, and realized that we had permanently altered the chemistry of the column with the surfactant. And when we went to do robustness on a new column, we didn't have specificity. So we had to redevelop the method on the new column. And anyways, it was a bit of a mess. It delayed the project quite a bit. So, and then there's forced degradation. Forced degradation is subjecting the drug substance and drug product to extreme stress conditions like acid, base, oxidative, which we use peroxide, temperature, and then also photolytic degradation, light degradation, to kind of accelerate chemical degradation. And you're doing a couple of things. One, you're looking for degradants in a general sense. And then two, we're also looking to make sure that the method is stability indicating. So a stability indicating method is a method where there are no impurities that are co-eluting with the main API peak. So you can imagine if you have some degradation happen and one of the degradants is underneath that peak, you're not going to see that degradation. You're not going to know that it's happening because it's going to be masked. So when you do the stability, the forced degradation, we're looking for peaks. We have to do mass balance to make sure that when we take the assay value and the related compound, total related compounds of a nominal preparation, and then we do the same with the forced degradation preparations, that they add up and we get as close as we can to 100% of the original amount in the nominal sample. So it can support an understanding of degradation pathways, but for this, in this case, in this instance, forced degradation is really to confirm method specificity and confirm the stability indicating nature of the method. All right, on to characterization, and this is specific to drug product characterization. At this point, you would have a method that is sufficiently developed, sufficiently specific for the impurities and the excipients that you're aware of at the time, has reasonable precision and accuracy and sensitivity. That's really where you want to get to. And you've got some sense of what you want your system suitability to be. And all of that is so that you're generating this body of data that you can rely on later. We know that it really, in the system suitability, the more you can generate, the more confidence that you have in your methodology. So for characterization, you want to look at the chemical, physical, and performance attributes of your drug product. And with that, you want to try to establish baseline variability. And this is hard when you're looking at lab scale lots of drug product. But if you have different raw materials, are you getting a different release profile? If you have, maybe it's a really low concentration of your active, and how consistently can it be made? How much variability are you seeing in that assay value? You want to look at the formulation robustness, and then ID those sources of variability in the drug product. There's no requirement to assess the edge of failure per the ICH, but it's a good idea to understand where the edge of failure is. So if you have a, kind of as an example, if you're looking at something that is the, that creates the consistency of like a topical product. So you've got something that forms the viscosity of the product, and maybe the specification range of the excipient is wider than what you can allow for your drug product. And so then you kind of have to look at your excipient specifications that's coming from the manufacturer versus what you really need. And it might be that you need to tighten that up. So things like that, you just want to make sure that you're not on the nice edge of something, especially something critical like release profile. A couple of the studies that we do during characterization is excipient compatibility, and that is taking the excipient and mixing it with the API and looking at how, if there's any chemical degradation, if there's any physical interaction, what happens there. Another one that we do is R&D stability, and that's typically the drug product and maybe the API as well, going up at accelerated conditions and looking at the degradation profile. So doing these early, excipient compatibility, you have to take with a grain of salt, because it's, sometimes you've got an excipient in there that is a really low concentration, and you're putting the API and the excipient together. It might not be indicative of what's really going to happen in the product, but it could give you a view into what could happen later. So you want to use both of these to support your formulation selection decisions. If you've got a formulation that has a lot higher, maybe there's a different excipient in it, and you've got a higher degradation pathway or degradation trend, then you might want to pick a different formulation. Early screening of your drug product also helps avoid stability surprises later. You certainly don't want this happening during a clinical study. And then also tells you what degradations that you can expect degradation products that you can expect to see. Another thing that you want to do during characterization is figure out your control strategy. And your control strategy goes beyond just specifications. It's the tests and the limits that go on the specification, but then it's also looking at how do these specifications dovetail together from your API to your excipients. The example that I just gave on viscosity of an excipient that goes into a topical product. You want to look at how all of this is controlling your product to get you to a consistent and safe product that's efficacious, right? So control strategy can include reduced testing. It can include real-time release testing. And it just helps you figure out how you're going to ensure the quality of your product. One of the big things that analytical development does is residual solvent assessments, and then also elemental impurity assessments. Residual solvents are the requirement for residual solvents is in the drug product, but most of the time we're controlling that in the drug substance and in the excipients. So for the residual solvents, the first thing that you want to do is go through and look at the residual solvent statements that you're getting for all of the excipients and for your API, and determine which solvents are used or produced in the manufacture of those excipients, and then which could be reasonably expected to be in there, right? And all of that should be provided by the manufacturer. And then you want to make a global assessment. So looking at the residual solvents, would they combine to fail in your finished product if they were at the limit? Then you have to develop a control strategy and say, based on what I know, we need to test these 3 excipients and our API for whatever solvents could possibly be in there. It's possible for you to do a risk assessment and say that this solvent is used early in the process and therefore wouldn't expect to be in the API or the excipient. You can also provide data that shows that you don't have those solvents. You know, maybe you've tested 3 different lots of the excipient or the raw material and you're not seeing them in there. And then you want to document that justification. Here are all the excipients. Here's the solvents that could be in there. This is what we're going to test, and this is how we're going to make sure that there are no residual solvents above the limit in ICHQ3C. Elemental impurities is a little bit different because elemental impurities can come from the drug substance and excipients. It can also come from the equipment and the packaging. So to start, you want to do something very similar, a lot of information gathering, get your elemental impurities statements from your manufacturers, and then possibly do some risk-based screening. So if you think that there could be impurities, elemental impurities in some of your excipients, you can test those and see if you're seeing anything in there. You can test product that has gone through the manufacturing process, test some packaging. You can even do an extractable study to see if any elemental impurities are being extracted. Once you have all of that, you compile it into a risk assessment. The risk assessment, in the end, you have to meet the ICHQ3D or the USP limits for the elemental impurities. And then they're different for different dosage forms, which is different from the residual solvents. The elemental impurities have different limits for whether it's oral, topical, inhalation, and parenteral. So you need to take that into account. You compile all the data, do your risk assessment, and determine your control strategy. Are there any excipients that need to be tested? Does the product need to be tested every time? And then you need to document the justification of that control strategy. On to method validation. And this is the only slide we have on method validation. We'll get into a lot more detail of method validation at another time, but there's different types of validation. So there's qualification, validation, and verification. Qualification is used in early stage development, maybe preclinical. If you're sending something for animal studies, it's an assessment of the method's performance. There's no requirement for a protocol or really acceptance criteria. And it's really focused on specificity. And but in this case, even in early stage development, it's really important that the method is well developed to the point where you can analyze for impurities. And the reason for that is that you could rely on some of those animal studies later for qualification of impurities. So the more information that you have early on, again, the better off you're going to be late stage development. It is I recognize that it's oftentimes you're going to have to do things more than once. It might be that you develop and qualify or validate a method more than once during the course of product development, and that's because you learn new things. But the value in having a good method overweighs the work that has to happen, maybe to revalidate if there's a formulation change. So validation is per ICH-Q2R2 and USP. It's predefined method, your system suitability that we talked about before, validation instructions, how to make your linearity solutions, how to perform your spiking studies, and then the acceptance criteria that's documented in a protocol. It is that you want to assess the specificity, linearity, accuracy, precision, and then sometimes sensitivity if it's a impurity method or something you're looking at low levels. And then you want to also, in your validation report, report your robustness data for that method, All of that can happen during development, and then also your force degradation from development. And then verification is for compendial methods. They're methods that have already been validated and have been published. Verification doesn't have to be a full validation. It can be sometimes, depending on your comfort level with it, or if you had any new impurities that were added to the method. But it really, what criteria you're checking in your verification needs to be determined based on the method and then also based on your drug product. And for verification, we typically assess precision. We do an N of 6, and then specificity of the method, as well as sensitivity for any impurity methods. All right, and then late development. Another thing that Analytical Development performs or manages with a contract lab is extractable and leachable testing. So leachables are chemical entities that migrate into a drug product from either the the manufacturing components or systems, packaging, or the delivery device components. And for a leachable, leachables are under the established manufacturing and labeled storage conditions. So this is the actual product. What actually happens in the product when it is in the packaging. And then an extractable is something where we're looking at the exact same thing, but under laboratory conditions. We're looking to see what can we extract from the packaging or the delivery device or even from manufacturing. There is the draft guidance, ICHQ 3E, that has come out, which is really risk-based assessment, very similar to the elemental impurities. And it focuses mostly on organic leachables, but does cover elemental impurities as well. And when you make some packaging changes, you need to look at the extractable leachables and see if there's any new risk that comes up from the change in that packaging. Again, the extractable leachable is risk-based, and this is a diagram that came out of there. So it looks at things like the manufacturing conditions, like is there high pressure, high temperature that makes it higher risk, or the route of administration, things that are injectable are a lot higher risk than like an oral or a topical, and then duration of treatment and things like that. And now stability testing. Again, this is extremely simplified, but just a quick overview of analytical development as it relates to stability testing. The first is a stability study design, looking at what conditions the product needs to be stored at. Is it a room temperature or a refrigerated product? Should we put samples up at intermediate conditions? Do we expect to see significant degradation? A lot of this is informed by your R&D stability testing that you've already done. Also looking at how many lots are going up, the packaging configuration, the orientation, those kind of things to design the stability study to support the shelf life of the product. Then also photostability, which is ICHQ 1B. Photostability looks at the degradation of the product from exposure to light. You're looking at UV and visible light. I think it's 1.2 million lux hours is one of them. I don't remember what the other one is. But you want to look at your product, your API, and your product in the packaging to see if your product does have a propensity for degradation. Does the packaging protect it adequately? A lot of the photostability information, you're already doing a lot of this in your force degradation, so you should have a good idea of what to expect. And then in-use stability, this is for any container or package that is multi-use. So something like a cough syrup or another oral solution, you should perform in-use stability testing over the course of how long somebody would reasonably be expected to use it. You want to finalize specifications in late stage. So these are your drug substance, your excipient, critical utilities like water or gases, in process or bulk specifications, your drug product release, and then your stability specifications. So once you get all of those finalized, you want to draft a justification of specification report that has information like batch consistency, stability data, excipient consistency, a lot of your, maybe some forced egg data in there, explanation of your impurities and how you're controlling. For this, go back to the previous one. We talked about this for about an hour. And then method transfers. So method transfers are covered under USP 1224, also a mention of it in ICH Q2R2. The method transfer, the extent of the work that needs to be done for the method transfer really depends on what the method is, the experience with the method at the receiving laboratory, and the comfort level. So it could be a full or a partial revalidation if the method's already validated. If the method hasn't been validated, it could be a co-validation where the laboratories are performing parts in tandem. So maybe you have the lab, both labs looking at specificity, method precision, and maybe some sensitivity. Maybe they're not repeating the accuracy portions. Or it's just a comparative analysis of representative samples. So sometimes your method transfer, if it's a relatively simple like assay method, can just be a analysis of 6 samples at the both of the laboratories, and you compare both the precision at the labs and then also a comparison of the value that was achieved at both laboratories. You can do that with pooled RSD or percent difference. And then in late stage is the CTD, which is Common Technical Document, CMC sections, the chemistry and manufacturing controls that are supported by analytical development. First is a quality overall summary in Module 2, which is a summary of all the sections that are down below. In the drug substance sections, 3 to S3 is where the specifications and the justification of spec go. Or no, that's where the elucidation of structure goes. And then control of drug substance or the spec goes, but then reference standards and stability. And then in the drug products, the control of excipients, control of drug product, all of your analytical methods for both the API and the finished products are provided, as well as the validation, your justification of spec, justification of residual solvent testing and elemental impurities, reference standard. Maybe if you did you had to do some extractable leachable testing, the container closure section would have that, and then stability with the stability data, the protocols, specification, and stability commitment. And then life cycle management. This is turning into a kind of a new topic. So anybody that's done, has gone through commercialization knows that there's always some changes. So it seems that what we're seeing in ICH and what we're seeing from FDA is a movement towards flexibility and being more nimble in making improvements to methods, making changes to specifications. Sometimes things happen and you don't have a choice, but other times you recognize that there's something that we could be doing better as you get more experience with the product, more experience with the method. So finding ways, if you're following ICH Q14, that you can provide a body of data that will allow you to easily make those changes is really good. And that's kind of all of the stuff that we talked about before with the characterization and the ATP, the robustness studies, all of those kind of things. Lifecycle management for AD is also supporting specification changes if there is a change in the impurity profile. Maybe you have a new degradant that shows up, things like that. Sometimes excipient changes have to happen. You can't get your excipient from a supplier that you had previously. You need to source a new one. And then also support for out of specification investigations, which hopefully never ever happens. But when it does, then analytical development usually is going back and looking at all that data that was generated to try to support that investigation. So I've got an analytical development checklist here. Kind of goes through everything that we already talked about. Data gathering, the compendial checks, API sourcing, impurity assessment, structural elucidation, setting your QTTP and your CQA to get your analytical target profile so you can develop your method developments, and maybe some qualification if you need that for preclinical studies. Identifying sources of your product variability, excipient compatibility, and R&D stability. I'm just reading all of these off. Developing a control strategy. The whole point that I want to make here is that, analytical development, there's a lot of it that goes into developing methods that are fit for purpose, developing specifications that ensure that the product that is manufactured is safe for consumption, that it is consistent, and that it is efficacious. I think that a lot of times in product development, hindsight is 20/20. You can look back and go, we really should have looked at more lots of that raw material. We really should have, all of those kinds of things. And going through this checklist and doing things this way helps to prevent the problems later. It helps prevent costly delays. So the conclusion, yay! AD is more than method development and validation. So you want to start by gathering all available information. Start with a really good base. It is just so exciting to make that first injection. Hopefully I'm preaching to the choir with a few of you here, but try to hold off just a little bit and do the work. Take a day or two to gather all of the information so that you have a good base, you have a good starting point, you really understand the method that you're supposed to be developing. Take advantage of the risk assessment approaches for things like elemental impurities and extractable leachables. Data generated and development really supports well-defined control strategy, and also can prevent you from testing things that really don't matter. Because one of the worst things is doing an out-of-specification investigation for something that has no tie to product quality. And then early analytical development will prevent later costly issues. So with that, I say thank you for joining us and see if we have any questions. So it looks like we had some that were emailed in before. How do you decide when to use a compendial method versus building a new one? If the compendial method isn't specific for all of your impurities, or if it isn't specific for the excipients that you're using, a lot of times the compendial method will be like a topical ointment, with whatever API. And depending on what excipients you're using in yours, you could have some pretty significant interference. So if the method isn't specific and if it isn't sensitive enough, that's usually when you need to build a new one. The next one is, how do you set raw material specifications when your suppliers are variable? First of all, find a new supplier. That would be the easiest way. And then you really are looking, when you're setting your raw material specifications, for a outlier, not an outlier, but you're looking to set it against kind of the worst case. Hopefully, if your supplier is a bit variable, it still would meet any compendial requirements, or it still would meet some like the ICH requirements for impurities. But if the raw material is that variable, then you really need to try to source something different. What are red flags? What red flags are there in early data that could predict regulatory questions? Number one is impurities above the ICH qualification limit. And then another is impurities above the ICH identification limit that you haven't identified already. So those are going to cause red flags. I think also a really steep degradation in unaccelerated, that you're going to have that. And then understanding your product, whether it is a suspension, making sure that you have particle size data, and that the particle size data that's used in your early clinical work is consistent with the particle size data in your later clinical work and what you're going to be putting to market. Have you ever had a method that worked in development but failed in QC? Yes, we have. More than I would like. Sometimes it's a change in excipient. That's why I would talk a lot about excipient robustness and making sure you're looking at multiple excipient lots. We've had viscosity methods that didn't work later because of a change in the excipient viscosity. We've had impurity methods where there's new impurities that showed up. One I mentioned was from packaging. We had benzophenone that came through from the label into a semi-solid. It seems like once you have a method in analytical development that is being run by the people that essentially birthed it, and then it goes to QC and it's run by people that are reading what you wrote and executing. And sometimes sometimes the work that you did and the understanding that the laboratory, the analytical development laboratory has, isn't translated into the test method. So I think that's where a lot of the robustness, looking at differences in instrumentation and things like that, really come into play. And that's it. Those are the questions that we have for today. Thanks again for joining us. Again, we're in about 3 weeks. We're going to be doing organic impurities, ICH Q3A and 3B, talking about the reporting limit, the identification limit, the qualification limits, the differences between drug substance, drug product, degradants, and process impurities. So I hope that you can join us there. Thanks.